it will be something that you've already seen, but I will put some special touches to it for you now. So remember, we were talking about cells have to do work, and to do work, they have to have some energy, and the energy production is usually the source of the energy. Short-term energy storage is in ATP, and we showed you the structure of ATP. But then I also told you that besides ATP, there are other compounds within the cell that may be driving some reactions also. So your ATP is here, and then there are some that are even higher than ATP in terms of their phosphoryl transfer potential, and therefore they are better donors than your ATP. So we look at these compounds, and these will come to play very soon. So watch out for these PEPs and PPGAs and acetyl phosphate and so on. Okay, so this is one way to drive reactions. The other way is, of course, by oxidation reduction reactions, right? That is another way to do it. And these oxidation reduction reactions always occur when you have electron carriers. So let's talk a little bit about electron carriers and what do they do. So remember, you have the glucose as your source of energy. Okay, you've given glucose to the cell. Now this glucose will have to be oxidized. The word oxidation will be used because glucose will be the donor for all these hydrogens that will be carried away by these electron carriers, and these electron carriers will eventually take the hydrogen, a proton and the electron to the electron transport chain, and through the electron transport chain you will get energy out of it again. Okay, so oxidation reduction reactions become very important in terms of using them for production of energy. So what are some carriers, as we call them, that assist in ripping off the hydrogen atom, proton and electron from there and take it to the electron transport chain? So remember, up to now, glucose was in the cytoplasm. Now these carriers will have to take those electrons and take it to the membrane where the electron transport chain is located. So one of the most common electron carrier that you come across in any of the system is the NAD. NAD is, of course, a nicotinamide adenine dinucleotide. So if you look at the structure, to which category of macromolecule is this? Does this belong to?
Nucleic acid.
It is like a nucleotide, it's a nucleic acid. Just like AMP, ATP, and those are also units of your nucleotides. This is also, so all the energy production seems to be located within these groups of nucleotides or some version of a nucleotide here, okay? So this has a nicotinamide, it has a ribose, and then it has a couple of phosphates, then you have another ribose, and then you have adenine. So this becomes your adenine, nicotinamide, adenine, you have adenine, and dinucleotide. So this is a dinucleotide. Sugar and phosphate is a nucleotide, right? So this is a nucleotide. So this is a dinucleotide. And this, the idea with this compound is that it is capable of carrying hydrogen. So you can see this hydrogen shown here is what it is capable of carrying the hydrogen at this point in nicotinamide. Whilst there is another electron carrier which is called NADP, which has an additional phosphate here. So NADP has the additional phosphate here. This becomes a different carrier. Again, it can only carry the electron or the proton up here, right? So NAD, NADP are two electron carriers. And I should clarify here for you that these two are very easily, they can exist in oxidized or reduced states. So when they carry the hydrogen, they can carry it, and then they will dump it back, come back without and carry more. So the reduced oxidized states will be are very common in NAD and NADP. Another thing that we have noticed that NAD is usually used in catabolic reactions where energy is to be means compounds have to be broken down. NAD is the carrier, but when it is a anabolic pathway or synthesis is occurring, you will usually see NADP as the carrier. So there is very fine distinction line between the two. Mostly NAD being used for catabolic activities or catabolic pathways. NADP will be used when we do a lot of anabolism or synthesis of molecules. So here are the reduced and the oxidized states being shown here. Is it going to operate for me? Yeah. So more electron carriers that you'll come across. FAD is a second one, which is a flavin adenine dinucleotide. It's again a dinucleotide. And there's one also close to this, is called FMN. This is the flavin mononucleotide. We also call it the riboflavin phosphate. So these two, of course, related ones also carry hydrogen on these two spots there, right? Then another carrier, which is called coenzyme Q, CoQ. It is a quinone and sometimes also called ubiquinone. And again, it is a hydrogen carrier. It is a carrier, electron carrier here, right? So again, these can be back into oxidized or reduced states. There are some more electron carriers that we want to describe right now. Cytochromes are a group of compounds that also are electron carriers. They actually use the iron in them. You can see the Fe in the center of it. Iron to use as their electron carrier atom here. So iron is actually a part of this heme group, this flat four ring structure. This is your heme, part of your hemoglobin also, as you would know, but in the this compound definitely heme is the one. Heme has the iron in it, and this iron becomes the electron carrier for this cytochrome. Whilst there is another electron carrier which we call ferredoxin, and not only ferredoxin, but there are others also that are non-heme iron proteins. That means they are proteins. They don't have the heme structure, but they do have iron as well as sulfur in them most of the time. But in this, again, your iron is being used to carry the electrons, but this iron is not part of the heme as in the cytochromes. So cytochromes and ferredoxins do differ in these characteristics. They both carry the electrons on the iron, but the iron in cytochrome is part of the heme, whilst in ferredoxin, iron is not part of the heme. Okay. So we talk about pathways. We talked about how reactions are connected together because the product of one reaction acts as a substrate for another, and the cell usually has all these metabolic activities created as a pathway. So it's easy for them to work through it. Now these pathways, which we could be anabolic or catabolic pathways. If they are both, then they are called amphibolic pathways. So some pathways will act as in terms of a catabolic activity, and some will act as anabolic. But some of our pathways that we're going to study today, they are considered to be amphibolic. They go both ways. So for example, when we talk about glucose glycolysis, glucose going all the way to pyruvate, this is a catabolic pathway. But the reversal of it is called gluconeogenesis, which is using most of the enzymes are same, except for about 4 different enzymes that will be needed that will replace the 3 enzymes in your glycolysis. So if you look at this pathway, this is clearly, you can see that when this green double arrowed means that enzyme is capable of reverse reaction also. But then there are some enzymes in a pathway that go in one direction only, and that's the only way the cell will be able to control which direction the cell is going, whether it is synthesizing or it's going to be breaking down that compound, right? So if you look at it, the pathways that we will study, and you've already studied these are your Embden-Meyerhof pathway. This is also an amphibolic pathway. We are showing it to you here. The pentose phosphate pathway, which we'll look at also later, is also an amphibolic. And even your tricarboxylic acid cycle, the TCA cycle, is a amphibolic pathway. So just establishing that the cell definitely prefers to have amphibolic pathways, so that some of the enzymes can be used commonly for synthesis as well as for breakdown, and entering a few exceptional one for each pathway is what we'll be studying now. Okay, so this pathway, amphibolic pathways, function both as catabolic and anabolic pathways is what we established. Now we talk a little bit about something called central metabolic pathways. Now, most living cells try to, use some pathways as their main pathways. So as such, glycolysis and TCA are considered to be the central metabolic pathways. Why? Because they generate 12 of the intermediates that are needed as carbon skeleton for biosynthesis. So through glycolysis and TCA, you will see most of the intermediates being used as carbon skeletons for biosynthesis. And therefore, it becomes easy for the cell to control these metabolic pathways. Heterotrophs that are using, remember, use organic compounds for their carbon source. They can use other carbon sources besides carbohydrates. We capitalize on working on carbohydrates, but the metabolic pathway for carbohydrates are definitely consist of the central metabolic pathways. That means when they use other substrates besides carbohydrates, these will also be converted into some of the intermediates of these two pathways. So if a bacterium is capable of hydrolyzing or breaking down petroleum, even that will be eventually, you will see that they will be able to bring it back into the central metabolic pathway. So definitely our central metabolic pathways become very important in terms of different types of microbes which can metabolize some fancy substrates, but they will eventually merge somewhere into the central metabolic pathway. So the word glycolysis, if you think of it, is nothing but to convert glucose to pyruvate. Glucose is a 6-carbon compound. It's going to be converted to a 3-carbon compound. So the term glycolysis is just breakdown of a 6-carbon compound into two 3-carbon compound. Now, what are some of these metabolic intermediates that we are talking about that will be used as carbon skeletons for biosynthesis, and where do they come from? So without knowing them now, because we haven't even looked at the pathways, just to give you an idea, so these are supposed to be your 6 important intermediates from Embden-Meyerhof pathway or the HMP or the TCA. These are all intermediates from these pathways that will be precursors to the synthesis of either polysaccharides, synthesis of murine. What is murine? Peptidoglycan. This is a special component of the cell wall of bacteria. So murine synthesis uses fructose 6-phosphate. Nucleic acid synthesis will use ribose 5-phosphate. Amino acid, lipids, fatty acids, all the precursors for these either your monomers for your proteins, or monomers for your lipids, or monomers for your polysaccharides, or monomers for even your peptidoglycan and nucleic acid. They all come from this carbon skeletons that are produced from this central metabolic pathways. Am I giving you the idea that these 12 compounds are very important, and we will be using them again and again? So you want to know where they are generated and eventually where they will be used as precursors to synthesize what other stuff that the cell has to make. So your central pathway does create these, as we say, 12 metabolic intermediates, those 12 metabolic intermediates, which will be very important for us. Okay, so let's go ahead and say, studying glycolysis, is there only one way to go and break down glucose and convert it into pyruvate? No, that's not true. There are more than one routes for glycolysis, and we'll look at the alternate routes today. Of course, the main one that you have studied and you know very well is the Embden-Meyerhof pathway. We call it the EMP pathway. And of course, the second one, which is the hexose monophosphate pathway, which is abbreviated as HMP. We have two new ones that will be introduced to you because these two are not found in your eukaryotic cells. Entener-Diodorf pathway, which is abbreviated as ED pathway, and another new one which is only operative in some bacteria is Phosphokitolase pathway. We call it the Pka pathway. Okay, so these two we will study first in details, and then we will see how these vary from the main ones and how different they are. Okay. So as I said, ED and PK, the last two pathways are unique to prokaryotes. They are not found in your eukaryotes. We do have some modified EMP in some bacteria. So we'll look at some modifications of the EMP, which normally operates in eukaryotes and prokaryotes, but some bacteria may have modified their EMP, and we'll look at those also. In one bacterium where we know the Thermatoga meritama, which is the bacterium well known, is in that you will find EMP as well as ED pathway running at the same time. So what I'm trying to say that these pathways are not exclusive at one time. In some microbes, you may find two types of them going at the same time from glucose to pyruvate. E. coli actually is an interesting organism. It will metabolize glucose. If it's growing with glucose, it will use EMP. But when it is using gluconate, it will use the ET pathway. So depending on what is being fed to the microbe, it has the potential to use more than one glycolytic pathways. So we'll discuss the Embden-Meyerhof first so that we have the model of it, and then from there we'll break it down. So this pathway, of course, occurs in the cytoplasmic matrix of most microorganisms. Even in your eukaryotic plants and animals, you will find this is occurring in the cytoplasm. This is not a membrane-bound pathway, so we are looking at it in the enzymes for it to be in the cytosol. And of course, this is the most common pathway for glucose degradation to pyruvate during aerobic respiration. So this aerobic respiration, where oxygen is required, the main pathway that operates in those cells is usually EMP. But it also functions in the absence of oxygen, so it is not requiring oxygen. It can function in both ways, but this is the most common one where your aerobic respiration is occurring, but it does. Now, there are two phases that we see in the Embden-Meyerhof pathway. I like to cut it into two phases for us. One is when you're adding phosphates to your glucose or whatever the intermediate is. So you're adding glucose in the first phase. That means you're providing energy into it first, and then oxidation and production of NADH and ATP will be in the second phase. So we look at it into two phases. So what is the precursor, or what is the substrate for EMP? What is the main compound that will be used as your substrate for this whole pathway? It's called glucose 6-phosphate. So glucose 6-phosphate in itself can be produced by different ways, and it can be utilized in different ways also. Glucose 6-phosphate can be a substrate for EMP, but glucose 6-phosphate is also a precursor for the biosynthesis of polysaccharides. When we go into the biosynthesis of polysaccharides, you will see a glucose 6-phosphate being used as a precursor. Many aerobic and enteric bacteria, they can transport glucose via the group translocation. Remember, we talked about group translocation? What kind of a transport is this? Is it an active transport or is it a passive transport? Group translocation. Come on. Is it active or passive? It is an active pathway because it needs a phosphate donor there, right? So it uses the energy given by PEP, right? And what is the main idea about group translocation? What is the outcome of it? Anybody? When glucose is transported by group translocation, what happens to glucose? When it crosses across the membrane, it becomes phosphorylated. So during the transport only you can do the phosphorylation. That would be group translocation, where your PEP will be the phosphate donor, and using this phosphotransferase enzyme that we've shown you in the past. So glucose can already be phosphorylated when it enters the cell. If not, glucose can also be transported without being phosphorylated, and then it can be phosphorylated using the hexokinase. This enzyme will be after the uptake, even though this will be taken up. The big G is for glucose, okay? So this glucose can be phosphorylated afterwards, or it can be phosphorylated during the transport. Another source of this glucose 6-phosphate, which is the substrate for your EMP, is the cell may have stored glycogen. The glycogen storage is there, and these glycogen molecules can be converted to glucose 1-phosphate using a phosphorylase enzyme, and then glucose 1-phosphate can be converted to glucose 6-phosphate, which is your substrate that you're looking for, right? So and this will be the mutase enzyme that will convert the glucose 1-phosphate to glucose 6-phosphate. So I'm just showing you different sources of glucose 6-phosphate into the cell, and another use of the glucose 6-phosphate for the biosynthesis of polysaccharides. Any questions? Okay, so here is the pathway that we are looking at and will be studying. You're starting with the glucose 6-phosphate. I keep moving this thing. I wish I could do that. Okay, so here is where you begin here. This is your glucose 6-phosphate, 6-carbon compound, already has a phosphate on it in the 6th carbon position. So the first step in this will be to convert it to fructose 6-phosphate. And then from fructose 6-phosphate, you're going to add another phosphate to it. So you're using an ATP here. And of course, the enzymes are all mentioned down here for what they are. And this 6-carbon compound, fructose 1,6-diphosphate, is an important intermediate because now it is capable of splitting into 2 3-carbon compounds. One is the dihydroxyacetone phosphate, DHAP, or glyceraldehyde 3-phosphate. So these two, and these are interchangeable also. So eventually they will, so this step, these couple of steps are all phosphorylation. So this is the first phase of it, where you phosphorylate first the glucose, and then you phosphorylate the fructose 6-phosphate to become a diphosphate. And then comes the reactions that are energy extracting. These are energy adding into the compound, and then now you are extracting it. And how do you extract it? You actually are going to rip off the hydrogen from these carbon compounds, and these hydrogen electrons will be carried by electron carriers. So electron carriers come into operation now, or play, where inorganic phosphate will be used also, but it will also take away the hydrogen electrons and put it on the carrier. So your carriers are coming into use now. And as such, these phosphates, which are now, this is supposed to be substrate level phosphorylation. This is called substrate level phosphorylation because your inorganic phosphate is being used from the cytosol and is being placed on your 3-carbon compound. And then once you put the phosphate in, once you take out the electrons, this next step will be the step where you will synthesize ATPs using the ADP here. And then we go on, and eventually you come to the last step where you take away again the second phosphate from it, and you come down with a 3-carbon compound, which we call pyruvate. Now, during this transfer, you can see that you come to a compound, which is a very important one, which is Phosphoenol Pyruvate. This 2-phosphorylated, this pyruvate, which has a phosphorus in it, is supposed to have a more energy potential to donate phosphate than even your ATP. And this phosphoenol pyruvate is the one that will be used during the transport of glucose when you are doing a group translocation. So this arm shows you group translocation. Glucose, as it is entering, is being phosphorylated by Phosphoenol Pyruvate and converting to glucose 6-phosphate. But glucose can be transported without being phosphorylated, and in this case, you will be using up an ATP to make it into a glucose 6-phosphate. So two mechanisms of glucose which is transported across, and whether it uses an ATP to phosphorylate it or it uses the PEP to phosphorylate it. You will come up with the precursor to this whole EMP, which is your glucose 6-phosphate. Do you understand this glycolysis and the cycle? Any questions? Now you will ask me, do we have to memorize all this? In most cases, you should understand the whole idea and what are the phases of it and what is occurring. Knowing the group of enzyme that will be used. For example, whenever an electron carrier comes in and picks up the electrons, you will see the enzyme called dehydrogenase. Okay, so kinase phosphorylases are the enzymes for adding of phosphates from ATP or taking it away. Whilst your dehydrogenases come into use when you have to take away electrons from there. Questions? Comments? Nothing? Are we okay with this information? Is it all that you've already studied, right?
Yes.
Yes. So am I just kind of reviewing it for you, right? That's all I'm doing. Okay, so we move on and see this pathway in a different book, which is much simpler to me. You're looking at the 6-carbon compound, which is glucose, and your ATP adding the phosphate to make it into glucose 6-phosphate. So your starting of the pathway starts from glucose 6-phosphate. It doesn't start from glucose here. So that's why you can see this being highlighted here as the compound that will go into the the Embden-Meyerhof pathway. This is still a 6-carbon compound, glucose to fructose, and then another ATP being added. So 2-phosphate being added to the two ends of your 6-carbon compound. This is the 6-carbon phase up to now, and then it breaks down and you get the 3-carbon phase coming up where all this oxidation will occur and where you will be drawing energy out of it. So looking at this picture, it shows very clearly that 2 ATPs are added and 4 ATPs are generated. Okay, so the net gain for your Embden-Meyerhof pathway is only 2 ATPs. So the cell has to put in 2 ATPs to draw 2 extra ones when it's converting glucose to pyruvate. So we call it the addition of phosphate, or we priming the pump here in the first phase, and then the oxidation step where it generates the reduced forms of the electron carriers. So high energy molecules are used to synthesize ATP, and this to also understand, please remember this is an inorganic phosphate. This is substrate level phosphorylation that is occurring in this where it will be used and eventually will be producing an ATP for us later, okay? So inorganic phosphates are being added here. So this is called substrate level phosphorylation. Another version of it, if you want to look at it, and if this agrees with you, again, glucose to glucose 6-phosphate, fructose 6-phosphate, the fructose 1,6-bisphosphate, then the two alternate ones, glyceraldehyde, phosphoglyceraldehyde, and dihydroxyacetone phosphate, and then again some steps of electrons carriers coming in and eventually coming down to pyruvate. And here is your very important compound called Phosphoenol Pyruvate. So if you want to summarize glycolysis, of course it is just glucose using up 2 ATPs and 2 inorganic phosphates and 2 NAD carriers will eventually give you 2 molecules of pyruvate. It will produce a net 2 ATPs and 2 carriers that are carrying the electrons. So this is a summary. Please remember this. You may be asked for that. Okay, so in some bacteria, such as in E. coli, it has been studied that when E. coli is under phosphate-limited conditions, that means there is not enough phosphate in the cell or in the environment, it modifies this EMP, and that is called the Methylglyoxal bypass. So Methylglyoxal bypass is actually a very dangerous path that they have to take, because phosphate is not found in the environment or in the cell. It's in a phosphate limited condition. Let's go back and see where the phosphates were being used. Remember, I told you that inorganic phosphate is needed at this substrate level phosphorylation, and if there is not enough phosphate, how is going to run the glycolysis is what we're going to look at by the Methylglyoxal bypass. So in this case, you know the two 3-carbon compounds produced at that step where the DHAP is there and the glyceraldehyde 3-phosphate is there. So methylglyoxal pathway uses an enzyme called methylglyoxal synthase that will actually liberate the phosphate. So it is actually releasing an inorganic phosphate. In your regular pathway, you were incorporating an inorganic phosphate. So actually it is releasing the inorganic phosphate in this case, and you come up with a compound which is called methylglyoxal. Now methylglyoxal is very reactive, and it destroys nucleic acids and proteins by reacting with guanine and adenine in your nucleotides. And with amino acids such as arginine, lysine, and cysteine, they are all very susceptible to methylglyoxal action. So this toxic compound is starting to accumulate in the cell because the cell is under phosphate limiting condition, and it is going through this step here. When this compound accumulates in the cell, various proteins are synthesized, including the membrane proteins that will excrete this out of the cell. And it will also produce an enzyme called glyoxylase, which is the next step. In the next step, this is the glyoxylase enzyme that will convert your methylglyoxal into a safe lactate, and then lactate will convert to pyruvate. So this is still a glycolysis pathway because it is ending up in pyruvate, but one of the intermediates in this is very reactive, and it has to be quickly converted into lactate and pyruvate, or it has to be excreted out of the cell. So under phosphate limiting conditions, E. coli can go through this alternate or modified EMP, as we call it. Now, whenever we study pathways, we are also going to be, because we are running these pathways in a living entity or a cell or a bacterium, we need to know where and how these pathways are regulated. So I'll mention the regulation as we go along to say what are the main enzymes that are regulated so that this pathway is operated under the best conditions and not overtaxing the cell. Okay, so this methylglyoxal synthase, which is the one that will produce the methylglyoxal, this enzyme number 1 as we call it, is regulated by inorganic phosphate through feedback inhibition. Now we will be talking about regulation, all describing these to you in a little bit in the next chapter or so, where you will learn a lot about feedback inhibition, feed-forward activation. Have you heard of those before? Have you heard of feedback inhibition at least?
Yes.
Okay, so you understand that there is inorganic phosphate will be used for feedback inhibition, and also it is activated by its substrate, which is the DHAP. So this DHAP is an activator for this enzyme, whilst the inorganic phosphate inhibits it. So definitely a very delicate balance is created in this production of methylglyoxal. This is one alternate pathway or modification of your Embden-Meyerhof pathway. Now, how is this main pathway, EMP, how is it regulated? What are the enzymes that regulate this pathway in bacteria? The phosphofructokinase, which is abbreviated as PFK, and Pyruvate Kinase. These are the two enzymes that do not catalyze reverse reaction, and this is due to thermodynamics, of course, but these are the only exclusive enzymes in your EMP that are not reversible, and therefore your EMP pathway will be regulated by these two enzymes. So what PFK is the main control for EMP. This enzyme also needs magnesium ions there. Now there are certain activators for this enzyme. So ADP and GDP activate the fructokinase enzyme. This is repressed by PEP. You remember this is one of the intermediate towards the end. So actually, if there's too much PEP, your EMP will be down-regulated, will be repressed. In yeast, this is in E. coli or your normal bacteria. In yeast, AMP activates the PFK. ATP and citrate represses it because it has a different pathways, different end products for that. Therefore, their regulators are different in yeast. Whenever you are trying to find out if your bacterium is using EMP or not, you look for this enzyme. If you can detect this enzyme in that bacterium, PFK, you know that this bacterium is using Embden-Meyerhof pathway. So this is definitely an indicator to detect EMP occurring in any cell. Of course, your substrate for this enzyme is fructose 6-phosphate. This is a very important compound in bacteria because it's actually a precursor of amino sugars and other polymers such as your murine. So murine, being an exclusive compound found in bacteria only, requires this pathway to be operative. So this intermediate fructose 6-phosphate is there, and this fructose 6-phosphate can be channeled into the production of cell wall component, the peptidoglycan. Now, the other enzyme which also controls your EMP is PK, which is your, of course, Pyruvate Kinase, which comes towards the end of the pathway. And this regulates the pathway by feed-forward activation. So in this case, what is it going to do? When fructose 1,6-diphosphate is accumulated in the cell, PK gets activated. Now, feed-forward activation is that the last enzyme in the pathway is activated by the substrate of the first enzyme. So the substrate of the first enzyme is your fructose 6-phosphate. So here, this enzyme will be activated to know that I am going to get my substrate because that enzyme is working or the substrate is there. So it will be activated by that. Let's look at it. Sorry. I just run through it. No? Let's go back. That will help. Okay, so where are these enzymes that we are talking about? So here is your enzyme number 11. This is your Phosphoenol Pyruvate. Now I should go even further down. Simpler ones. Where is it? Here it is. So here is the enzyme, which is one way. You can see this enzyme, phosphofructokinase, PFK is here, and pyruvate kinase is here. Now, these two enzymes are only one way. Of course, hexokinases. The hexokinase is also a one-way enzyme, but there is an alternate enzyme, which is the phosphatase, which is used during gluconeogenesis. But these two, when we start off from the glucose 6-phosphate, these are the only two enzymes that go in one direction. So these are the controlling enzymes that will control the pathway. Now this one, a pyruvate kinase, is activated by feed-forward. So if the substrate is present here, this substrate fructose 6-phosphate is present, then this enzyme gets activated because it knows now that this pathway will run and I should be ready for converting the phosphoenol pyruvate to pyruvate. So kind of giving you an idea as to how this whole operates. I'm much better this way so I can go back easier. Okay, so we were talking about the regulation and how feed-forward activation occurs for PK. Okay. So moving on, looking at it, so if you would think that I called your pathway, Embden-Meyerhof pathway, as an amphibolic pathway, so why am I calling it an amphibolic pathway? Because we've seen glucose being broken down to pyruvate in this pathway, but this pathway can be reversed very easily and become an anabolic pathway. That means a synthesis pathway, because the gluconeogenesis, which is almost parallel to it, with most of the enzymes that used the same, this becomes an anabolic pathway. So glycolysis is also an anabolic pathway, meaning glucose can be synthesized. So if you're looking at it, glycolysis anabolic, because intermediates from this glycolysis are being used for synthesis of different amino acids or so. So here are some of the precursors for amino acid. Fructose 6-phosphate goes into amino sugar synthesis. Glucose 6-phosphate goes into polysaccharide, pentose, and aromatic amino acid synthesis. Phospholipids are created from DHAP. Serine, glycine, cysteine, these are amino acids that are synthesized from phosphoglyceraldehyde. And your PEP, of course, is used for aromatic amino acid synthesis and for the muramic acid also. So you can see that this pathway generates those intermediates that will be used for synthesis. Therefore, glycolysis is considered as an anabolic pathway also. And you can see some of its regulation that occurs. So when I talk about regulation, in these figures, you should be able to read that this enzyme that converts fructose 6-phosphate to fructose bisphosphate or diphosphate, its positive regulator is ATP and its negative regulator is PEP. You understand the figure? Because in the exam, I will give you figures and tell me what are the regulators and how do they work and on which enzyme. So being able to read these figures is very important to you, not to memorize all of it, but to know some of the ways that these pathways are regulated. So here is another enzyme that is the Pka enzyme, and here's the PFK enzyme. The Pka enzyme is being positively regulated by what? This fructose bisphosphate. So you know this is a feed-forward regulation, was this was a positive and a negative regulation on the enzyme called PFK. So I've kind of summarized what we had looked at and given you the lowdown on how glycolysis can be an anabolic pathway, because it generates all the intermediates that will be used for synthesis of some very important amino sugars and amino acids and all other compounds. And its regulation is being shown, the Embden-Meyerhof pathway, by just those two enzymes are the most critical ones that will be regulated. Now coming to the process called gluconeogenesis, this of course is needed. Why? Because sometimes you give the cell no glucose, but you're giving them something else to go on, like fructose, mannose, or galactose, or you're giving them pyruvate in itself. So the cell may have to synthesize or reverse this pathway to go up to glucose. So as such, many microbes can grow on alternate sources also. So glucose, fructose, and mannose are gluconeogenic. That means they will be using these directly from these to convert it into glucose. Galactose, when it is synthesized, is a nucleoside diphosphate derivative. So it does not get converted directly, but it is a nucleoside diphosphate derivative. And we'll talk about the nucleoside diphosphates very soon. Bacteria and algae synthesize glucose, glycogen, and starch from adenosine diphosphate. So again, adenosine diphosphate glucose will be needed to create the large forms or the storage forms of glucose into the cell. So gluconeogenesis is always synthesis of glucose and its more advanced form of compounds from there. So when you look at glycolysis and gluconeogenesis, they look almost as if it is a functional reversal. So gluconeogenesis is just a functional reversal, but the two pathways are not identical. So we cannot say that they are totally identical. 7 enzymes are shared by the two pathways, but 4 enzymes are unique to gluconeogenesis. So you'll need those 4 extra enzymes to be able to reverse this pathway. So it's not a truly reversal pathway of glycolysis, it's a functional reversal. So gluconeogenesis being shown here again, and you know this forward one was using PFK and Pka enzymes. Those were unique to your glycolysis. Now the 4 unique enzymes for your gluconeogenesis are pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose bisphosphatase, and glucose 6-phosphatase. So these are exclusive enzymes that are operating in your gluconeogenesis. So now we'll see how is gluconeogenesis regulated. This is actually through the control of fructose phosphatase activities. So where is that fructose phosphatase enzyme? Here is the fructose phosphatase enzyme. So one that is almost parallel to the PFK in your glycolysis, this reversal enzyme is the controlling enzyme here. Now this enzyme looks at the energy status in the cell. If the cell, this enzyme will be activated by ATP, and then your supply of carbon skeletons will be created for growth. And if it's only AMP and ATP, of course, are very important in knowing the energy status. So if it's AMP is present, then it represses the enzyme activity when the energy status is too low for growth. So it won't allow growth or storage of these glucose polymers later. So this enzyme is activated by ATP and it is repressed by AMP. Does that make sense to you?
Yes.
Very good. Okay, so we come back to our 12 intermediates that will be used in synthesis. Now that we've studied EMP, we can look at what are some of those that we had looked at in this pathway that will be useful now. You know, glucose 6-phosphate was the start one. Fructose 6-phosphate is the second derivative or the intermediate in this pathway. So the first one is used as a precursor for polysaccharide. This one is used for murine. And we go down to more than. In the triose phosphates, the EMP generates it, and this is used for lipid synthesis. Phosphoglycerate, also produced from EMP, used for amino acids. PEP, generated by the EMP, also for amino acid synthesis. And pyruvate, of course, in itself, as a final product of your EMP, is also used for amino acids. So I'm trying to put into your head that these pathways, why are they called central metabolic pathways? Because they generate these intermediates that will be used for the synthesis for a lot of other organic compounds in the cell. We move on to the HMP now, which is your also called the pentose phosphate pathway. Now, this is a very important pathway which runs parallel to or at the same time as EMP, because there are some intermediates that are not provided by your EMP. And if you look at the previous slide again, you can see these two ones, ribose 5-phosphate and erythrose 4-phosphate. So this is a 5-carbon compound and this is a 4-carbon compound. These are not generated during the EMP, and the cell needs it for nucleic acid synthesis or amino acid synthesis. So this pathway can run at the same time as EMP needs to be run, because these two compounds, intermediates, are generated by HMP only. Okay. So the proportion of how much glucose will go through EMP and how much it will go through, we will look at it in the end of the chapter, and we'll look at it which cells run more of this and less of this, because this is not needed at that time, or so on. Okay. So this pathway can operate at the same time as your EMP, as I said, and it can also operate at the same time as Entner-Diodorf pathway, which we will come to next. This pathway also can operate aerobically or anaerobically, and it is again an amphibolic pathway. So this is the general properties of it. Now we can break down this pathway into 3 steps, very easy. Just like we did 2 steps for your EMP, we break this down into 3 steps for you. Oxidation of glucose 6-phosphate, remember your precursor, of course, the main one that will enter into the HMP or EMP is glucose 6-phosphate. So oxidation of glucose 6-phosphate will occur at the first step. So this is using the enzyme dehydrogenase. Whenever you have, remember when you are taking away electrons, oxidation is occurring and you need the enzyme dehydrogenase. And a special enzyme called lactonase is being used. So just remember, dehydrogenase and lactonase are 2 enzymes that participate in the first step of HMP. The second step will be isomerization of this ribulose 5-phosphate using isomerase and epimerase enzymes. So in this second step, converting the 5-carbon compound into different isomeric or epimeric forms will be occurring. And finally, the main part of your pathway is transformation of the 5-carbon sugars to generate 3-carbon, 4-carbon, 6-carbon, or 7-carbon compounds using 2 important enzymes we call transaldolase and transketolase. When it is a transaldolase, the aldolase transfers 3 carbon fragments, whilst the transketolase transfers 2 carbon fragments. I suppose this is if it is too much repetition, just let me know, then I will run faster than this. But if it is useful to go slower like this, then I can continue at my pace that I'm doing now. Please let me know which one to go for.
At the pace you're going now.
Is it okay?
Yeah, thank you.
Thank you. Because some of you may not have touched on these pathways for a while, and it's good to review it. And the perspective for us is very different from your general chemistry or biochemistry classes. This is, we are creating a living organism is what we are looking at. So regulation is also very critical as such as to how does the cell regulate these pathways. So I'm adding a new touch to your biochemistry that you have studied up to now. So anyway, so as some of the intermediates of your HMP, which are going to be very important, are your ribose 5-phosphate and your erythrose 4-phosphate. Remember, the 5-carbon and the 4-carbon ones will be needed for nucleotide and aromatic amino acid synthesis. And also, in this pathway, the only one where the electron carrier is NADPH. Now, what does this indicate? That this pathway generates your electron carrier, which can be now used as reducing power during biosynthesis. So this will be used during anabolic activity. So this is only generated of all the pathways that we have. This is the one of glycolytic pathways is what I'm saying. This is the only one that generates NADPH, and this will be used for biosynthetic synthesis activity. So 3 important intermediates from the HMP, which are important in the general cell regulation. Oh no, this doesn't go this way then. Okay, so what is this pathway? As I said, we start with the glucose converting to glucose 6-phosphate, the same idea, either through active transport or during group translocation. Your glucose 6-phosphate is your first step. From here, glycolysis begins. The first step in this pathway is 2 enzymes acting here, the lactonase as well as dehydrogenase. So here it is, the glucose 6-phosphate dehydrogenase and lactonase, number 2 and 3 enzymes are here. So what are they doing? The electron carrier that comes in is NADP, not NAD, and it is generating your reduced form of your electron carrier. As well as it has converted it from a glucose 6-phosphate to 6-phosphogluconate. So whenever you hear gluconate as an intermediate, you know it is the HMP, the pathway operating, not glycolysis. Gluconate is created by your enzyme lactonase. So lactonase, gluconate, if you can relate together, product and the enzyme. And of course, your dehydrogenase is operating at the same time and taking away your electrons from here. Then the next step is also very unique, in which this is going to be a dehydrogenase as well as you are taking away a carbon dioxide. That one carbon is being lost here. So from a 6-carbon compound, you're coming out down to a 5-carbon compound. So two important steps in which both of them are dehydrogenases, but in this case, converting it from a glucose to gluconate, and from here, from gluconate, you're taking away a carbon and converting it into a ribulose, which is a 5-carbon compound. Okay, so this is the first series of it, where you are actually using dehydrogenation reactions and converting your carbon compound from 6 to 5 carbon. Now comes the second series of reactions, where your ribulose 5-phosphate you have created can either be using an epimerase to go towards xylulose 5-phosphate, or using a isomerase to convert it into a ribose 5-phosphate. So this is just interconversion by epimerases and isomerases. And then the last, which is a whole pathway where it is interconversion, where you will be putting together different carbon numbers and generating the other carbon 4 and 6 and 7 and 8 will be created. And this is definitely a very important interconversion where transketolases and transaldolases will be used. Okay, so this pathway, if you look at it, what does it end up with? So if you want to look at that, and we'll come back to this last bit of it as to how it is regulated. So here is your pathway shown in the proper way. So hexose, it's called the hexose monophosphate pathway, HMP, and you start with glucose 6-phosphate. This is the phosphogluconate, ribulose phosphate. Then you go into the epimerase and so on, so that you have xylulose 5-phosphate and ribose phosphate. Those are the two required to go into interconversion. So you have two 5-carbon compounds coming together. Transketolase will convert it into a 3 and a 7-carbon compound, and the 3 and 7 can come together and use the transaldolase and make it into a 6-carbon and a 4-carbon compound. So all these conversions can occur during this pathway. And again, you come back to the transketolase action, and what have you generated? Glyceraldehyde 3-phosphate, which is a 3-carbon compound that is also common with your EMP, and here you are generating your pyruvate. So the whole idea, glucose going to pyruvate, is in this occurring. But during this conversions, you are creating some intermediates that will be drawn away for other synthesis of other amino acids or nucleotides or so on. And of course, you can do the glyceraldehyde 3-phosphate going back to fructose 6-phosphate and regenerating also. So as such, the important thing you look at is the sugar transformation reactions, and then produce sugars that are needed for biosynthesis, and then sugars that can be further degraded and be converted to pyruvate. So 4 important ideas into your HMP. Here is another version of it, if you, not a version, but it is the sugar transformation reactions. You don't need to memorize, but just to able to understand these figures, where 5 and 5-carbon compounds are coming together to make a 10-carbon compound, which is then being split up into 7 plus 3. Okay, so there's no 10-carbon compound, but 5 and 5 we can redistribute by using the transaldolase or ketolase, and you will produce the 7 plus 3. Here is 5 and 4 coming together, xylulose and erythrose coming together to give you 6 plus 3, which is fructose and glyceraldehyde. And your transaldolase reactions, these are both transketolase reactions. These are transaldolase reactions where the 3 carbons will be transferred. Here 2 carbons are being transferred. Here 3 carbons are being transferred. And your 7 plus 3 come together to give you 6 plus 4. So we've generated quite a few varieties of carbon compounds that can be used somewhere. Now, looking at it, the last statement here is important to us here. So how is this pathway regulated? Now, if you look at the pathway, there is only one enzyme in this pathway. Let me go back to this picture again. Not here. There is only one enzyme in this pathway that is unable to catalyze the reverse reaction, and that enzyme is lactonase. So that lactonase enzyme, this is operating here, is the one that goes in one direction. Where was it in which picture? I could show you this. But anyway, just remember that lactonase is the only enzyme, but this is not the enzyme that controls the pathway. The regulation of the pathway is exerted through glucose 6-phosphate dehydrogenase and phosphogluconate dehydrogenase. So the two dehydrogenases, that is your enzyme number 2 and enzyme number 4, which I've highlighted here, these are the controlling enzymes for the pathway. This pathway is inhibited when too much of NADPH is accumulated. So negative regulation with accumulation of this is also another way of controlling, even though lactonase is the enzyme that is non-reversible. Oh, here it is, probably. So here we're showing it in an easier way if you want to look at it or not. It doesn't matter. I give you the various versions so that. So here is a summary of HMP. Glucose 6-phosphate plus 12 NADP will be used, and 7 water molecules, and you will generate, if you go through the TCA cycle also all the way, then it will be 6 carbon dioxide, 12 NADP reduced forms, and inorganic phosphates also. So as we said, HMP can operate at the same time as EMP or even ED. Since EMP cannot supply all the intermediates, and NADPH generated from this is needed for biosynthesis. HMP is a major glycolytic pathway for bacteria that uses pentose sugars or do not have any other glycolytic pathways. They can just run on HMP and they will be happy. So if they are fed only pentose sugars, this will be the major glycolytic pathway that they will use. Now let's look at a modification. This operates in Brucella abortus. Brucella abortus is a bacterium that lacks a functional EMP or ED pathway, so it has to run only on an HMP cycle. So it uses an oxidative HMP cycle. And how different is it is what we're going to look at real quickly. So glucose being converted to glucose 6-phosphate, entering into the HMP, the same step where you're creating the gluconate and then ribulose 5-phosphate. This is the same pathway HMP uses regularly. But here is the variation where 1-glyceraldehyde 3-phosphate will go into the fructose 6-phosphate, and because these intermediates, because it does not have a functional EMP, so some of the intermediates that we needed that were coming from the EMP are now being generated by the oxidative cycle of this HMP, which will give you this very important intermediate fructose 6-phosphate is coming up. So this definitely is an important part of this oxidative HMP cycle that has to operate if there is a non-functional EMP and the cell does not have an ET pathway. Moving on to Entner-Diodorf pathway, which is new to you. And this pathway, of course, is used by mainly by soil bacteria and a few gram-negative bacteria. We found it only in some of them. And this actually replaces the first phase of EMP. So there is just, I would say it's a modified one where you're using EMP, but later on, not from the beginning. So let's look at ED pathway. So if you look at the ED pathway, you start with the glucose 6-phosphate. You go to the gluconate, which is your HMP intermediate. You go to something that is called 2-keto-deoxyphosphogluconate. And this is a unique step, which is creating, we call it KDPG. So the KDPG is being created, which is new in this Entner-Diodorf pathway. And the enzyme that is unique to this pathway is the 6-phosphogluconate dehydratase. So this enzyme number 2 is the one that is unique here. And of course, the next one will also be unique because you've created a unique intermediate that you haven't seen in EMP or in ED pathways, or EMP or HMP. So this is found in ED, which is the aldolase. This aldolase will create pyruvate and glyceraldehyde 3-phosphate for you. Okay, so this unique intermediate, which is the KDPG, keto deoxyphosphogluconate, KDPG. So this, if you detect this, you know the cell is running the ET pathway because this is unique to this pathway. But the first step in it is similar to your HMP, and the rest of it from on to go to pyruvate will definitely be your regular EMP pathway. So it can go into this regular EMP pathway and give you the pyruvate, or it can use this enzyme and give you the pyruvate here. So definitely, if you look at it, it looks like a hybrid to me. Entner-Diodorf pathway starting here. The reactions that are similar in the HMP pathway first, then the unique step of creating KDPG, and then glyceraldehyde 3-phosphate going into the conversion to pyruvate, and this will be your regular EMP pathway. So you're looking at HMP here, unique step, and then EMP here. The yield becomes a little less here. In the Entner-Diodorf pathway, you only yield 1 ATP, and you also generate 2 different types of electron carriers, NADP, one of them, and one NAD. So definitely the outcome is very different by these very unique bacteria that are inhabitants of soil, and they use this Entner-Diodorf pathway. And here is another version of it. You can look at it. It's the same thing. I don't want to go through it. So let's look at some bacteria and say, where does which bacteria substitute or use ED instead of EMP, or they use it together, both together at the same time. So you are familiar with some names like Bacillus subtilis. Bacillus subtilis is your regular bacterium that uses EMP pathway, and it does not have an ED pathway. It's a gram-positive bacterium anyway. ED will probably be found mostly in your gram-negative bacteria. So Escherichia coli, you are familiar with, and other enteric bacteria, they may have an ED as well as an EMP pathway. But then there are some which are very fancy bacteria that we are going to be talking about more and more so, such as Rhizobium japonicum and Thiobacillus ferroxidin and Xanthomonas faecioli. These have nothing but the ED pathway. They don't have the EMP. Okay. Even your very important one, which we will talk about very soon, is Ralstonia eutropha, which we used to call Alkylogenes eutrophus, also has the ED pathway. So these are some special bacteria that are using the ED pathway, and they are not using the EMP. Okay? The Halococcus, no, not the Halococcus, the Halobacterium, the Clostridium aceticum, and the Sulfobus, we will talk about these bacteria very soon also. They have the ED pathway, they don't have the EMP there. Now we talk about some bacteria are capable of oxidizing the glucose extracellularly also, and then transporting it. Kind of interesting idea. So which are these bacteria? When glucose concentration is high, some strains of Pseudomonas will oxidize the glucose at the periplasmic region. So within the periplasm, we are talking about, this is of course a gram-negative bacterium, so it has a periplasmic region. This glucose dehydrogenase and gluconate dehydrogenase are cytoplasmic membrane quinoproteins containing the PQQ. So these enzymes are located in the periplasm, glucose dehydrogenase and gluconate dehydrogenase. So actually you're going to start oxidation of the glucose right at the periplasm only. So it's called extracellular oxidation. That means it hasn't entered the cytoplasm and it's being oxidized right at the membrane level. Okay. Simple again, glucose dehydrogenase will produce the gluconate. The gluconate dehydrogenase will produce the ketogluconate, and that will be transported. So all these intermediates or the beginnings will be entering into the cell and will be joining your ET pathway, or they will be joining your EMP pathway. Okay, so the reduced PQQ is important because it's transported through specific transporters. This PQQ transfers electrons to the cytochrome C, and gluconate and the 2-ketoglutarate are transported through very special transporters that are needed here. So definitely, once it enters the cell, it is metabolized the same way as your ED pathway, or if it is coming here, it will go into the EMP pathway. So just to tell you that some bacteria of Pseudomonas, when they see high concentration of glucose, they go into all these different ways of transporting it into the cell to be used as energy sources. Okay, so this is a greedy bacterium, I would say. We go to the last pathway that we have to describe really quickly now is the phosphokitolase pathway. We call it the PK pathway. And this is normally found in very special bacteria, which are gram-positive. Lactate is the sole glucose fermentation product in the Homo fermentative lactic acid bacteria. There's a group of bacteria, we call them lactic acid bacteria. So this lactic acid bacteria convert your glucose to lactic acid as their product. Whilst the heterofermentative bacteria, which can produce more than other than just lactic acid, they can produce acetate and ethanol as their other end products. So there are two kinds of lactic acid bacteria, ones that are called homofermentative, the others that are called heterofermentative. And these two bacteria that we will describe that have the PK pathway, these are heterofermenters. Okay, so the Leuconostoc masteroides or the Bifidobacterium are heterofermenters, and these are supposed to be very good bacteria. If you ever look on your probiotics, these microbes are definitely part of your probiotic therapy, where you put good bacteria into your gut and your digestion is very healthy then. Actually, you will find these Bifidobacterium, these bacteria, Bifidobacterium bifidum, is mainly one of the microbes that is found in breastfed babies. So breastfeeding allows this bacterium to be in larger numbers, which gives a healthy microflora for that baby. So we have noticed that these two bacteria are very important in your gut bacteria composition. So we usually add those bacteria in your probiotics also. Okay, so what does it do? So Leuconostoc masteroides is a heterofermenter lactic acid bacterium. That means it not only produces lactate from your glucose, but it also produces acetate and ethanol through a special pathway which we will call PK pathway. So it has one PK for the xylulose 5-phosphate. So you're going to go from glucose to xylulose and then into your acetate and ethanol. Okay, so Leuconostoc masteroides. Whilst your Bifidobacterium produces lactate and acetate by the bifidum pathway, and this has two of these enzymes, phosphokitolase enzymes. One is for the fructose 6-phosphate, and the other is for the xylulose 5-phosphate. So they have the same xylulose 5-phosphate enzyme, phosphokitolase, that acts on this, but this one has an additional one. The phosphokitolase will act on the fructose 6-phosphate also. So here we go. The pathway is being shown where you are starting with the phosphokitolase enzymes that are being used in this pathway. So your glucose is coming through and you come up to the xylulose 5-phosphate. Remember, this is part of your HMP. And this xylulose 5-phosphate is target for this phosphokitolase, and this phosphokitolase will take it into a path where the ethanol will be the end product, and the bifurcation here will give you the lactate. So this is the enzyme, the phosphokitolase pathways for glucose stop, which has this one phosphokitolase only, which will end up in production of lactate as well as ethanol. Please don't get bogged down with all the intermediates or all. The whole idea for us is to look at the pathway, where it begins and where it ends. So being able to analyze this figure, you're looking at your glucose going through your HMP, producing your xylulose 5-phosphate, and this is your the enzyme. The substrate for this enzyme is xylulose 5-phosphate, and then producing ethanol and lactate as your two products. Okay, don't need to worry about all the enzymes. We are only capitalizing on the phosphokitolase and what its substrate is. This is the bifidum pathway. So here you will use 2 phosphokitolase.